my Wi-Fi goes, or if you can't see something. Can you see my cursor?
Not yet.
Oh, okay. I don't know if it's possible to. I'm going to do a laser pointer. Can you see the laser pointer? Maybe I'll share my entire screen. Maybe that will be better. I'll try that.
Sorry, we're not seeing presentation, but PowerPoint.
Say again, Carlos?
We're seeing PowerPoint with the notes and the slide design.
Oh, okay. I'm going to try something else. Hold on. Because I went into presentation mode. Okay, I'm going to try something else. 1 second. I have to go back to, right, stop presenting. Now I'm going to try again. Sorry. I'm not very good with Google Meet. I'm better on the other ones. Okay, present now. I'm going to do my entire screen. And then I'm going to say allow. Oops, window or screen to share. Entire screen. Okay, allow. Right, okay, now I'm going to go to PowerPoint. And I'm going to go to, okay, can you see my presentation without the notes and everything?
Yeah, awesome. And
Okay, everything. Okay, I can't see you guys, so just tell me if something goes wrong. Okay. So thank you very much again for the invitation. So I'm going to talk about some emerging applications of QSM, which is short, of course, for quantitative susceptibility mapping and much easier to say. And of course, we're talking here about magnetic susceptibility. So here's just a little bit of an outline of what I'll try to cover in about half an hour. So just tell me, yeah, okay, just about 5:05. I'll try and keep an eye on the watch. Okay, so I'm going to talk about what is QSM, just a tiny taste of how we do it conceptually without going into detail, a few of the advantages, and then I'm going to talk about some applications that we've looked at, mostly in the brain. I know you guys are technically a cardiac center, but I'm going to talk mostly about brain things, although obviously there's some vascular stuff in there, so cardiovascular. So obviously it's all very relevant. Okay, so magnetic susceptibility mapping. What is it? Why do we want to do it? And how do we do it? So as I'm sure you know, magnetic susceptibility is an intrinsic property of materials, including tissue, and it tells us how our tissues are going to behave when we put them inside the field, inside the scanner. So that's an official definition. This is a figure taken from the Schenck paper. And you can see that there's this spectrum of susceptibilities. When the susceptibility is negative, we call it diamagnetic. When it's positive, we call it paramagnetic. And in biological tissue, we don't really encounter anything that's ferromagnetic that keeps its magnetism after we remove it from the scan. Okay, so here's just a schematic diagram of the kinds of susceptibilities that we see in vivo. We can see that calcifications have a diamagnetic susceptibility. Everything's relative to the susceptibility of water or soft tissue, and we'll see how that comes about later. And then, of course, we have all the paramagnetic susceptibilities, which include things like deoxygenated blood and liver with iron overload and some deep brain tissues in the brain. Okay, so one way I like to think about susceptibility is using something called Wiedemann's additivity law, which tells us that the susceptibility of a tissue, or of anything really, is a mixture of the weighted sum of the susceptibility of each component. And so that really gives us this idea that susceptibility depends on tissue's chemical composition. And so if we think about the primary sources of susceptibility that we find in biological tissue, that automatically kind of leads us to several different applications. So for example, if we think about the major susceptibility source that most people know about is tissue iron, usually in the form of ferritin, so stored tissue iron. And this means that we can apply QSM to looking at deep brain iron-rich structures. We can target structures for deep brain stimulation. We can look at changes in iron in MS lesions, in Parkinson's, in Alzheimer's, in sickle cell anemia. There's also deoxyhemoglobin, as I mentioned, is highly paramagnetic, which means that we can start to look at veins, deoxygenated blood and veins. We can look at hemorrhages, microbleeds, and even functional QSM, which I'm not going to talk any more about. Myelin also has a specific susceptibility, so we can start to look at demyelination in different diseases like multiple sclerosis and Alzheimer's, calcifications. There's many, many more applications. And of course, microstructure and anisotropy, which again, I won't have time to talk about. So just a tiny bit of the background, which I'm sure many of you are very familiar with, but just for those of you who are not. So susceptibility leads to magnetic field perturbations. And if we put a cylinder or a sphere of a different susceptibility in the scanner, then we can see that they make these very nice kind of cloverleaf dipolar patterns of field perturbations. And if we know the susceptibility of that enclosure, of that sphere or cylinder, then we can very easily calculate these field perturbation patterns by convolving this susceptibility distribution with a unit dipole pattern, which looks like this. So this is the convolution operator. If we knew the susceptibilities, then we could very easily calculate the kinds of field maps or phase images that we would measure in the scanner. So that's all very well. We can think about a kind of toy example. If we have a numerical phantom or a simulation of a susceptibility distribution in the human head, then we can convolve that with this unit dipole function and calculate a field perturbation which looks something like this. And then if we actually convert that into an MRI phase, which we can measure, which is the quantity that we're going to measure, then that would look a little bit like this. So the main things to notice here are that we have these very, very large field perturbations around the air spaces in the head, which have a very positive susceptibility with respect to tissue, and also that those turn into wraps when we look at our field maps. And then there's just some equations for those of you who like equations down the side here to just show what's happening. So the phase differences that we measure in a gradient echo sequence are going to depend on this underlying susceptibility distribution in a convoluted manner. Okay, so just what is phase and how can we actually get some kind of measurement that will allow us to measure susceptibility? So in gradient echo MRI and T2 star weighted images, we have this magnetization vector here in the transverse or complex plane, which precesses around in this plane. So this precesses around at the Larmor frequency, and 99% of the time we're looking at the magnitude of this vector. That's what gives us our magnitude images. But then we can also look at the angle that this vector makes in the imaginary complex plane, which gives us our phase. And of course, we can create phase images, which look a bit like this. And we can see, as we saw before, that the phase is linearly dependent on these field perturbations that we're interested in, that depend on the underlying susceptibility distribution. Okay, so that's all very well, but the phase, if we just were to use phase images, the phase contrast, those phase images, the contrast in them is non-local and dependent on the orientation of the structures in those images. So the phase is affected by the tissue geometry and orientation with respect to the main field. And we can see an example of that here. We've got a cortical layer that's perpendicular to the field and one that's parallel to the field. And we can see that they have almost opposite contrast in this phase image. And the same thing here, if we rotate a fixed rat brain, then the contrast between the grey and white matter changes dramatically depending on the orientation of those structures with respect to B0. So that's the orientation dependence. Then we also have this non-local contrast. So here we have a magnitude image of the red nuclei and the substantia nigra, and we can see that the phase contrast spreads out around those structures, far from those structures themselves. So that's this non-local dipolar contrast. So ideally, we want to get to a susceptibility map that will not suffer from these problems. So how do we do that? Well, how to calculate susceptibility maps from phase images. First thing we have to do is acquire these T2 star weighted gradient echo images. And then these points seem very simple, but I think it's always worth mentioning it because the number of times that people come to me and say, yeah, I've got lots of data, can you do some QSM for me? And I say, well, where are your phase images? Where's your complex data? And they haven't saved it. So the main thing is to make sure that you save that phase data. If you're doing a study, don't throw it away. The scanner will automatically save it. Then these days, of course, we're all acquiring data with multi-channel RF coils. So you need to make sure that those data are combined properly. I believe that your workhorse scanner is probably a Philips, so it's probably doing a good coil combination using a sense kind of algorithm, so you don't have to worry too much. And then we have found that it's much better to collect multiple echo times and fit the phase over those echo times, or fit even the complex data over those echo times, to really get a good estimate of those magnetic field perturbations that we're really interested in. Okay, I won't say any more about that, about the methods, just, well, now how are we going to continue towards calculating the susceptibility? Well, the first thing we notice is that we have these wraps in the phase image, these jumps, these phase jumps, and we need to remove those by doing something called unwrapping, which I'll explain in a moment. The next thing we need to do is remove the very large field perturbations that arise from the air spaces in the head that we're not interested in. And so we're going to remove these background fields and just reveal the local fields that are caused by the underlying subtle tissue magnetic susceptibility changes. And finally, we have to solve that inverse problem. So go from the measured phase to the underlying susceptibility, do a susceptibility calculation and give a QSM, a susceptibility map. Okay, and just to mention that because we've removed these large-scale background fields, then the susceptibilities that we get are relative. It doesn't make them any less useful. It just means that they're not absolute susceptibilities. So we can't necessarily compare them very easily with a value that we would get, for example, if we put a piece of tissue in something like a squid magnetometer, so an independent way of measuring susceptibility. But it doesn't matter because the differences between susceptibilities in different tissues should still be the same. Here's just a bunch of different papers and reviews for anyone who's interested in finding out more. But I'm sure you can ask Christian and Carlos for those references if you're stuck. Okay, so the first step, just to recap, we need to acquire our images, and this is a multi-echo gradient echo sequence, which is kind of the workhorse for QSM, although of course we could use EPI or many other T2 star weighted gradient echo imaging sequences. And what the images look like, we've got our exponential decay according to R2 star at the top, and then we've got our linear increase in the phase with echo time here illustrated on the bottom. The next step, as I mentioned, is to unwrap and get rid of those phase jumps. So here we can see that the cause of those wraps or phase jumps shown here is that this phase is defined as an angle, and every time it crosses this imaginary axis, it jumps by 2 pi radians. So we don't quite know how many times we've gone round, and so we have to basically put back those 2 pi phase jumps using something called an unwrapping algorithm, of which there are many, and get this nicely unwrapped phase image. In fact, our group has actually invented 2 phase unwrapping algorithms, which we've published. One is called Segue, which is a region-based algorithm. So it's based on kind of growing out regions and looking at the boundaries of those regions and at the wraps and adding on appropriate integer values of 2 pi to unwrap the images. That was developed by Anita Carser, and it's a lot faster than the gold standard region-based algorithm Prelude, as you can see here in a few of these examples. So for a standard 3 Tesla brain image, Prelude might take about 16 minutes on a standard PC, and Segue takes about 28 seconds. It's also a MATLAB implementation, very similar concept to Prelude. And the reason we developed it, the reason Anita developed it, is because she was fed up of waiting about 10 hours for a head and neck image to unwrap. So Segue can do it in about 10 or 11 minutes. And similarly, we've got speed up for pelvic images too. And you can download it for free here. Or just email us if you can't. Just Google Segue UCL and you'll find it. And we've published that in 2019. So more recently, and this was just very recently published in September, Barbara Demerska from our group has come up with this time a path-based instead of a region-based unwrapping technique. It's called Romeo. It's based on the Julia programming language. Again, freely downloadable, and it's really fast and very robust. And it can also be used on multi-echo or multi-time point imaging, so that it can be used for 4-dimensional unwrapping as well, and it's very reliable. You can see how it does a little bit better compared to both the Prelude Gold Standard and other best path-based techniques as opposed to region-based techniques. And here you can see a histogram of errors or some 3T data, both for Prelude and best path, and then for Romeo, which does a lot better. So smaller integer numbers of errors in Romeo. And this is a logarithmic scale on the histogram. And again, that's freely downloadable. You can see the MRM paper. Okay, so the final step, how do we calculate our susceptibilities? How do we solve this ill-posed inverse problem? Well, oh, sorry, background field removal. I'm not going to really talk much about that. We exploit the harmonic properties of the fields due to the sources outside our region of interest. And there's a really good review if you want to read more about that. You can find that in Schweizer, in NMR and Biomedicine in 2016. I'm not really going to talk more about that. The method that we tend to use for most of the studies that I'm going to show you is the PDF method, or the projection onto dipole fields method, which exploits this property here. Okay, so the final step, as I mentioned, we can exploit this phenomenon. I'm just going to explain the very conceptually simple method of QSM just for those who've not seen it before, but I'm sure all of you have. So here, if we express our convolution in the Fourier domain, so if we Fourier transform everything, of course, it becomes a very nice multiplication, and then we can express the inverse problem as a division, and we can see that it's ill-posed. It doesn't have a unique solution because the Fourier transform of this dipole function has a cone of zeros in it at the magic angle, which means that we run into problems. So a very, very simple way of solving that problem. So here's that cone of zeros in the Fourier transform of the dipole function that I mentioned before. If we just run that naively, there's the cone of zeros, we get a mess. But it's one thing we can do, which is the first thing we thought of back in 2009, is to simply truncate this function at a particular value, maintaining its sign. And of course, then we had to figure out which value to truncate it at. And of course, we now know that there is an ideal value that we can truncate it at, which is 1.5 or 3 or 2/3, depending on how you express it. And we can then correct for any underestimation of susceptibilities that you can start to see creeping in here. So that's so-called truncated case-based division or thresholded case-based division, which is conceptually very, very simple, computationally simple, but things have moved on since then, as they should have done. So now most people tend to use these regularization-based methods, and of course the regularization can be done either in case space or image space. Case-based methods don't really use any prior information, small iterations, but when we use these more sophisticated image-based methods, then we do input this prior information, smoothing, iterations. And this figure is really just here to show that we have to be careful what prior information we supply, because if we, for example, supply a missing spatial prior here, a missing inclusion, then we do get something. We know there is a susceptibility inclusion here, but it doesn't have the susceptibility value that it should have. And if we put a deliberately cubic spatial prior in here, deliberately wrong one, then we get a cubically shaped output. So just a little note that we need to be careful that if we're adding prior information, we need to be careful that it's actually the right prior information. And then, of course, we've had our first QSM challenge to try to come together as a QSM community and really decide what is the best QSM method. So everybody submitted their favorite methods. There was a winner, but the consensus was that it was very, very smooth and cartoon-like. Some of our submissions did quite well and scored high on the metrics, but maybe had a little bit too much noise. So now, of course, there's been QSM Challenge 2, which I know Carlos is in the process of publishing. So we'll see, how far we've progressed, because one of the problems was we didn't have an ideal ground truth. So this Cosmos method, which was a multi-angle acquisition, doesn't produce an ideal ground truth. But now we have perhaps a real ground truth. Of course, everything's moved on very rapidly, and of course the deep learning methods have starting to take over. So these are just a few of the early deep learning networks that have started to be used for various stages of the QSM pipeline. There's a little review if you want to have a look at those, and this is going to expand and expand as time goes on, and we're starting to dip our toes into these methods as well. Right, what are the advantages of doing this QSM pipeline? Why do we want to do this? Well, it's really, really efficient. We get a lot of bang for buck, so we can get a lot of multiple contrasts from a single acquisition. We get our standard T2 star weighted data, we get our phase images, we can fit our magnitude data to get an R2 star map. We can create susceptibility weighted images by doing some processing and multiplying the processed magnitude and phase data together, and the radiologists really like that. It has really made it into the clinic because it amplifies phase changes like microbleeds and things like that. And then, of course, we can do our QSM processing and get a susceptibility map, which overcomes the disadvantages of those phase images and the SWI images. So it overcomes the orientation dependence and non-local contrast. So here's an example showing how that non-local contrast is resolved and overcome in the susceptibility map. So here you can actually see the increased susceptibility in the red nuclei and substantia nigra. And then here's an example of how the orientation dependence in this piece of fixed brain tissue, which you can see very dramatically in these two phase images at 90-degree orientations to each other and with respect to the main magnetic field. So if we take a profile across all these different structures here, so we've got some white matter structures, some grey matter cortical layers, and if we take a profile across those, then we can see that actually the phase contrast is almost opposite, but the susceptibility map resolves that very nicely, and the contrast is almost identical. So let's get on to some applications. That was a very rapid tour. I don't know if, I'm hoping that everyone can hear me okay and that everything's all right. I guess we'll have questions at the end, but if you do have anything burning, then feel free to chip in now. Everything good?
Everything is fine.
Okay, good, thank you. All right, so here we go. So I'm going to talk about healthy brain aging and cerebral amyloid angiopathy. So we have applied our pipeline, and we're constantly optimizing it in a very unique study of a birth cohort. So this is 500 people born in 1946, I think within a month of each other in March 1946. So they were all, well, 70, they're now about 74. And they have had both amyloid PET and a whole battery of MRI, because they're in the MRI PET system at UCL for about an hour. So you can imagine how many different MRI exams we tried to squeeze into that hour. It's a very large study with lots of different collaborating groups, but of course you get in about a 2-minute QSM sequence, and that's being repeated after 2 years. So the idea is that we hope to find MRI biomarkers that may even substitute for the amyloid PET and show us who in the cohort has become amyloid positive. And this is because these are healthy people. There's going to be a certain percentage of them that will become amyloid positive during that 2-year period. So we want to see really if QSM and other technique, MRI-based techniques, can provide biomarkers for dementia. So that study, I think they're almost finishing scanning. I don't know what's been happening during COVID. I believe they might be trying to continue scanning, but I'm not sure. So here's an example of some of the images that we've generated for that study. Of course, here we've got the magnitude and phase images, and here's the SWI that the radiologists like for looking for microbleeds. And then we've got our QSM here on the far right. The nice thing with QSM is that it can, unlike phase images, magnitude images, it can distinguish, and even SWI, it can distinguish between paramagnetic areas or microbleeds and diamagnetic calcifications, which we can see quite commonly here, for example, in the ventricles, sometimes at the midline of the brain as well. And what we have here is something called true SWI, which is meant to be a compromise between SWI and QSM. So it's meant to employ the advantages of both. So what we've got is we take the QSM map and invert it and process it and multiply it with the magnitude image, so that we amplify the susceptibility changes but get rid of the non-local and orientation dependent contrast that's in the SWI. So that's the idea behind true SWI. Hopefully the radiologists will like it. And the idea, we wanted to have a look at which of these images depict microbleeds most precisely. And we've actually tried to get some radiologists to do some microbleed counting to try and show which images are best for looking at microbleeds, and also that they can be distinguished from the calcifications. So yeah, as I said, we've tried to display the images to account for what the radiologists like looking at. And we did the same thing in a cohort of patients with cerebral amyloid angiopathy. So this is a buildup of amyloid in the vessels. And here are the results. You can see here that there was a lot of microbleed broadening and duplication in the filtered phase and SWI images. And here we have quite a large hemorrhage at the surface of the cortex. And here's a whole host of microbleeds over here. And what I hope you'll see, which I'm going to blow up in a minute, is that actually the doubled microbleeds, or there are microbleeds that appear in the SWI image that are not found in the QSM map. And that is because they're actually doubled because the extra microbleeds are not really there. They're part of that non-local dipolar effect, which is removed by the susceptibility mapping. And you can see that hopefully here, where here, for example, you can see the sort of doubling of the microbleeds that you get in the SWI that is not present in the true SWI and the QSM it is based on. So so that was shown by Barbara. And so the QSM is obviously going to avoid that kind of doubling or broadening of those microbleeds. Okay, we've also been collaborating with some Parkinson's disease clinicians who have already published some work looking at Parkinson's disease, and this time looking at a voxel-wise comparison of susceptibility changes in the cortex and in the deep brain grey matter. And what they found was that the susceptibility was greater in Parkinson's patients than controls, not only in the deep grey matter, which is very well known, like in the substantia nigra, pars compacta, but also in different cortical areas, as you can see here. So quite extensive increases in susceptibility in the cortex there. And this was done using a study-wise template. This study-wise template and paradigm or protocol was developed by Julio Acosta Cabronero, who I know you've worked with in the past. This study was also really interesting because it showed that the susceptibility was co-varying with lower cognitive performance. So increased susceptibility suggested that areas of increased iron content were associated with lower cognitive performance in these Parkinson's disease patients. And those associations were found in the hippocampus, the thalamus, putamen, and caudate. So quite extensive areas of association there. And that's been published by George Thomas et al. In 2020. Okay, so what about arteriovenous malformations? Well, my PhD student, ex-PhD student, Emma Biondetti, looked at these, and as we know, QSM can measure venous oxygenation. So there's a very nice, strong linear correlation between the deoxyhemoglobin concentration in blood and its susceptibility, which you can see here. And so we can use susceptibility to really probe oxygenation directly. And you can see these lovely maps from Audrey Fan very early on. And this is the basis of many applications from venography to QSM. And we wanted to use this to look at brain arteriovenous malformations. And you can see an example of one of these over here. And what AVMs are, is really where the arteries and veins are directly connected without a capillary bed through a very tortuous network of vessels called a nidus. And the idea is that we would find more oxygenated or arterialized blood in the vessels and the veins that drain the AVM, although this hadn't really been shown in vivo non-invasively before. Now, these AVM patients have an increased risk of stroke, hemorrhage, seizures, and other neurological problems. And so the idea is that if we can get physiological information in vivo about the oxygenation in these draining veins, then we can understand whether the treatments that are used to restore normal blood flow actually do restore normal blood flow and oxygenation. Of course, this is based on the deoxyhemoglobin content, as I just showed you. And this is our question: can we use susceptibility mapping to measure the higher oxygenation in these AVM draining veins? And if we look here, you can see that the answer is yes. So we use these very carefully drawn region of interest, both in the superior sagittal sinus as a control venous region, and also in the AVM draining veins outlined over here. And you can see that the draining vein oxygenation from the QSM measurements was significantly higher than the superior sagittal sinus measurements. And you can see that, yeah, here we go. So 85% oxygenation in the draining veins and about 75% oxygenation in the superior sagittal sinus. And again, this may help us understand whether the treatments have worked. And it could even help with automated detection and things like that. So one problem is that these oxygenation values don't necessarily agree with what you see in the literature from PET studies. So there's work to be done in absolute calibration, but the relative difference is very, very clear and consistent and significant. Okay, we have a very long-standing and extensive collaboration on sickle cell anemia, and so we've got some interesting results with QSM in those cohorts. So sickle cell anemia is a genetic disorder that affects hemoglobin, and in sickle cell anemia, the deoxygenated red blood cells change into this abnormal and fragile sickle shape. So it means they can't deliver enough oxygen to the body's tissues, and it causes several different kinds of symptoms. You get hemolytic anemia, you get vasculopathy, so the vessels get blocked, and sometimes that leads to bleeds. There can be clinical stroke. There can be these silent cerebral infarcts, which I'll explain a little bit more about later. And this can lead also to cognitive impairment. And as we know, brain iron is really important in cognitive function, and iron overload in older patients and in older diseases, as we saw in Parkinson's disease, can accelerate neurodegeneration. We're looking at the young end of the spectrum. These are mostly young patients, but we want to exploit the fact that the QSM is directly proportional to either tissue iron content or vascular oxygenation, and use QSM to investigate whether the sickle cell anemia affects brain iron levels or venous oxygenation. And this study looked at about 86 sickle cell anemia patients and about 25 healthy controls at 3 Tesla. And these were recruited from 2 clinical studies that you can see here, both the prevention of morbidity in sickle cell disease and the sleep asthma cohort study. So looking first at iron content in the deep brain regions here, in these different regions of interest highlighted over here, we found significant increases in the susceptibility in the red nucleus in the sickle cell anemia patients. So the rate of accumulation was much faster in the patients than in the controls. And this was specific to the red nucleus when we did this region of interest analysis. And Russell, the student who's been working on this, is actually also in the process of looking at some whole brain comparisons as well, using a similar kind of approach, using a voxel-wise template as I showed you for the Parkinson's study. So we know, the mechanism is unclear because there weren't any correlations between the susceptibility and the hematocrit, or the presence of silent cerebral infarcts. So we're not quite sure what the mechanism is here. If we start now looking at the venous oxygenation, these results are hot off the press. I just got them from Russell today. I had results for fewer subjects, but this is now from 88 sickle cell patients and 30 healthy controls. And Russell drew some very careful regions of interest. In fact, he's redrawn them since, so you can see there's a slight error here, but he's drawn some very careful regions of interest in the superior sagittal sinus in both the sickle and control subjects. And what you see here is a small but highly significant difference in the oxygenation between the sickle cell patients and the healthy controls. So overall, we get this tiny difference in this tiny decrease in oxygenation in sickle cell anemia. It would be really nice if we had an automated segmentation method for this superior sagittal sinus region of interest. We've got issues with trying to get rid of background fields because we're right on the edge of our region here, on the edge of the kind of support area where we have signal. So it's tricky, but these results are really robust. We've seen these with 2 different QSM pipelines, and we're confident that this is a small but significant difference. Here you can see that in a smaller group of patients with a different sequence, we don't actually see a significant difference, but we do see a trend in the same direction. These results were actually used to look at the venous oxygenation, comparing our QSM methods with the kind of, believe it or not, gold standard method, which is T2 relaxation under spin tagging. Now, the trust estimates are based on 4 voxels in the superior sagittal sinus. Our regions of interest have between 500 and 1,000 voxels. So you can already see some of the issues that we're facing here. The other issue with trust is that it depends, the results you can see depend really strongly on the type of hemoglobin which is used in the calibration model to convert between the T2 measurements that you make and the oxygenation values that you calculate. So if you use bovine hemoglobin or sickled hemoglobin or normal hemoglobin, you get really different results, as you can see here, between the controls and the sickle cell anemia patients. So only in the case of using a model with sickled hemoglobin do you get a significant difference between the oxygenation in the sickle cell anemia, superior sagittal sinus, and that in the controls. And this is kind of unusual because you've got actually an increased oxygenation in the sickle cell anemia compared to the controls. So although these are very, very variable, these actually do agree with results in the literature. And I would like to suggest that the result that I've just shown you is probably what we should be using, again, because we've got so many more voxels, and it's really robust, and we've proven it now in a very large cohort of patients. So this variable agreement between, what I haven't shown you is that the susceptibility and trust oxygenation measurements were moderately correlated in controls, but not in the sickle patients. So we obviously need more work to see what's going on with the trust, but I think we should ditch trust and use QSM, although it's going to be hard to persuade the hematologists and the people working in sickle cell anemia. Okay. And finally, looking at these silent cerebral infarcts. So what are these SCIs? They're white matter hyperintensities that don't correlate with any focal symptoms. So very different from a clinical stroke where someone's got a clear deficit, functional deficit. You don't see anything in their clinical presentation, but when you look at their images, you do see these white matter hyperintensities. We don't know what is actually inside these SCI lesions, whether they're edema or bleeds or anything. And so we really want to use QSM to have a look, and also to see if the susceptibility of these lesions varies as a function of whether people have sickle cell disease, how well they perform cognitively, their age, where these lesions are found in the brain, etc. So we've used several different QSM sequences in different subsets of these subjects. We've used a multi-echo gradient echo very standard sequence with a 1.15 millimeter isotropic resolution. And we've also used a series of multi-parametric mapping sequences, or MPM sequences. And Russell's shown that if you average the susceptibility values from these high resolution 1 millimeter isotropic resolution sequences, then you can actually reduce the noise in your susceptibility maps. And he presented that at the ISMRM this year. And these are what the images look like. So you can see here's our standard QSM, if you like, and here's the average of these 3 MPM QSM maps, and they look very similar, although this one's higher resolution. And what's really I think amazing is that we get this incredibly strong correlation, highly significant correlation between the susceptibility measured using the MPM sequence and the susceptibility measured using the standard SWI multi-echo gradient echo sequence, even though they've got different acquisition parameters, different echo times, different resolution. So this gives me great hope that we have a very robust pipeline here. And there's a lot of controversy, and there's a belief in the QSM community that you really need to use a reference region or tissue to calibrate all of your susceptibility results against. And I would argue that actually, as long as you use the same pipeline, then you really don't have to do that, because you get the same result, even if you use different acquisition sequences. So we're currently doing these correlations. Russell is working very hard to get these results ready for his PhD thesis, and I would say watch this space. Hopefully we'll be able to try and submit something for the next year's ISMRM on this. Okay, finally, and I know I'm running out of time here, but I'm just going to quickly whiz through, if I've got 5 more minutes, do I?
No problem. Please
okay,
continue.
Thanks. I'm going to quickly whiz through some results on epilepsy lesions, on focal cortical dysplasia. Okay, and this is work that's been done in collaboration with a lot of people who I'll show you at the end. But this is about the fact that about 50% of children who have drug-resistant epilepsy have these focal cortical dysplasia lesions, or FCD lesions. And surgery is only effective if we can actually identify these lesions. And it's surprisingly difficult to identify these lesions. They appear very heterogeneously. They sometimes lead to cortical thinning or thickening or blurring of the gray-white matter border. So it's very hard to identify these lesions. So we really thought, let's try and see if we can use QSM to identify the lesions. And because we have surgical resections, some of these children have surgical resections, then we can do histology on the tissue that's taken from them. And there are different classifications of these different lesions, these focal cortical dysplasias, and different types of cells that we find, but we also find calcifications, which, as we know, are diamagnetic. So we thought, well, why don't we try and use QSM to identify some of these lesions? And the hypothesis here is that the changes in susceptibility in these lesions would be associated with tissue iron content and calcifications as measured using an independent technique, a histological technique. So these are our cohorts. There were 40 patients with radiologically defined FCD lesions. They ranged from 2 to 18 years old, and 25 of them had resections with histology. So here was the MRI protocol, 3T Siemens Prisma, the 3D GRE sequence with 1.15 millimeter isotropic resolution, so the same one I showed you before. Here's our QSM pipeline, so multi-echo fit, Laplacian unwrapping, and Laplacian boundary value background field removal, and then an iterative Tikhonov regularization technique. So here are some of the susceptibility maps that we got in various different patients from this cohort with different types of lesions. And you can see here there's a very clear what we think is a calcification. I'm going to show you a bit more evidence for that later. And then some very heterogeneous radiological appearances for all the other different kinds of lesions, including hippocampal sclerosis over here. So what did the postdoc do, Sarah do? Well, we actually manually masked out the lesions and then extracted all these cortical surfaces at very different depths from the peel surface to try to measure some QSM profiles through these lesions at different depths. And this is what we got. So here, this is actually averaged over several different lesions, resected lesions. And these are results, susceptibility results in vivo. And you can see that you've got actually this kind of peak in susceptibility in the homotopic regions, so the control healthy regions on the contralateral hemisphere. And you can see that those are significantly different susceptibility values at these depths of 2 or 3 millimeters from the surface. So looks like there's a breakdown of the normal cortical structure in these lesions, and there's a decreased susceptibility in the lesions. So this peak only happens in the healthy homotopic regions. Oops, sorry. So what did we do? We compared these susceptibility profiles with histology on paraffin embedded tissue, and these were taken not only for staining but also for synchrotron radiation, X-ray fluorescence, to try and get a real elemental breakdown of what is in these tissues that could be causing any QSM differences, any susceptibility differences. And so these 2D maps at very high resolution were acquired in very thin slices. As you can see, here's an example of an iron map. So here, for example, we can see this region here with a decrease in the susceptibility value. We can see the cellular staining over here. And if we look at that region in the histology, we can see that there's a clear increase in the concentration of calcium and zinc in that lesion, especially compared to the normal levels of calcium and zinc in a healthy region over here. And we can also look at the myelin stain. So there's a decrease in myelin and what looks like a kind of a decrease in iron staining as well in this region compared to a healthy region of white matter over here. And you can see the quantification from the XRF over here. So what it looks like is that the susceptibility is sensitive to alterations in cortical structure in these FCD lesions, and the reductions in susceptibility in the lesions were associated with decreased iron content and also increased calcium content. And we also observed this increased zinc concentration, and zinc is also sort of strongly diamagnetic, so it ties in quite well. So it seems that the susceptibility mapping has a role to help identify and classify these FCD lesions. And the fact that we've got these resections happening in these epileptic patients provides us a unique opportunity to understand the susceptibility changes by comparing them with histology. So these are all the team involved in that epilepsy study. I'll just do a very quick summary of what I hope I've shown you in this, sorry, too long, 45 minutes. Susceptibility, as you can see, can be calculated from gradient echo phase images using QSM, which overcomes the non-local and orientation-dependent contrast in phase images. Many clinical applications are emerging based on the sensitivity of the susceptibility to the tissue mineralization, iron content, myelin and deoxyhemoglobin content. QSM can be used to look at microbleed burden and distinguishes bleeds from calcifications. QSM increases with cognitive severity in Parkinson's. QSM shows that AVM draining veins have higher oxygenation than the superior sagittal sinus. It suggests increased iron in the red nucleus in sickle cell anemia and oxygenation changes as well, which I didn't have time to put in the summary. It reveals abnormal zinc, calcium, and iron levels in epilepsy lesions, and it's going to get increasingly important because it reveals multiple pathophysiologically important tissue components. So I think it's going to be increasingly clinically applied. Now, this was my team the last time we could actually be physically together, so a long time ago now. This is my current team about a week ago, and this is the way we now talk to each other, as I'm sure as we're doing now. And these are all many of the collaborators who were involved in all of this hard work, and lots of funding that I'd like to acknowledge. And thank you very much. I'd really like to take any of your questions now. Okay, I'm gonna stop sharing. So let's stop. There we go.
Thank you very much, Karin, for the interesting talk.
Thank you. Sorry, I went over time. Okay. If you've got any questions, I'd be happy to talk to you.
Yes, there's a couple of questions. Cristian Montalva.
Hi, Karin. Thanks for your presentation. Hi, nice to meet you. I'm Christian. I'm the MRI technician in the lab, and I have a question about QSM. Do you have any experience about acquiring abdominal QSM?
A little bit. So we've processed some abdominal QSM, but we didn't acquire the images just for us. So they were actually acquired, and we used the data. It was actually a 3-point Dixon technique. So it was something that was being done anyway for a different study. It was something that if I had more time, I would show you. We were looking at bone marrow in the pelvis, and we have a project which, one of my other postdocs is working on, which is QSM in the prostate. So we hope to get very good at abdominal QSM, but we had the chance to develop it.
Yeah, the reason is we are with Carlos. We have a project. We have patients. They have an adenoma, but do you know the adenoma? They have the hypofunctional adenoma, and there are a patient they develop some more hypofunctional and hypofunctional. So the difference between them, they produce an iron particle. And the reason is with Carlos we acquired this abdominal QSM. But we discussed the thing. It's what happened to acquire the QSM in abdominal images, because we didn't know if we need to acquire the image in one breath hold or in a lot of, because the intestine movement, I don't know, they changed the phase. So we tried to change, and the spatial resolution, it's way different to the brain.
Yes.
So I don't know.
It's very challenging. So I can tell you a few things. I mean, I think obviously you're going to have bowel gas as a problem, and I think that the patients for the pelvic and prostate scans that they do for other clinical MRI scans, they prepare them carefully with something called Buscopan, which I believe removes a lot of the bowel gas before the experiment. And then in terms of breath hold, I can't remember now whether these images that we had were acquired during a single breath hold, but if you look in the paper by Bray and Carso, which I can send you later, then you can check whether they were breath hold or not. But I know that this is a real problem, and it's very challenging.
Yeah, I will email you for the paper. Thank you.
Yes, yeah, no worries.
Thank you. Carlos Castillo, do you have another question?
Yes. Well, first, are you hearing me? First?
Yes, thank you. Yes.
Yes. Okay. Good presentation. I wanted to ask a very technical question, because in QSM, the best of my knowledge, you assume that you have the same T for all the samples in the case space. That could affect the resolution or the interpretation of the data afterwards?
So I think what you're asking me is, does geometric distortion and dropout have an effect on the susceptibility map?
I'm not sure if
it's a spectrum, right? So you always have, a kind of a spectrum of error on the right, whether it's you can think about it as assuming the same echo time, or the fact that you assume that causing very slight distortions, and in extreme cases you would get signal dropout, right? So that's something that we're interested in and that we'd like to investigate, but we haven't really looked at it yet. So I think it is something that we should worry about, but the effect would be subtle. Yeah.
Time for only one another question. So I have a question about. You showed us some experiments in cortical areas, and when you do so, you have a kind of compromise when you do your PDF or whatever you're doing for background removal, because you need to take away a few pixels during the erosion process. So how far from the actual external area are you when you do that, and how you cope with this erosion process?
It cut out a bit, so I didn't hear if you were asking about epilepsy or the Parkinson's. It doesn't really matter, but I'm not sure.
Basically, you showed us two experiments when you.
Yeah, okay. The cortical. Yeah, no, I know. Yeah, the background field removal. So you're asking really about the masking and the effect on the cortex and the background field removal, right?
Yeah.
Yeah. So yes, it depends. Obviously, so masking is the kind of the hidden step that is very non-trivial in QSM, because as you said, it strongly affects the results that you're going to get from your background field removal and the susceptibilities you will get at the edges of the brain. I think that we in the study that we did, not the Parkinson's but the epilepsy, I think we used Laplacian boundary value background field removal to try and reduce how much we had to erode the brain by. So I think we tried to minimize that. And I think that also the reason we used that method is it was a little bit more robust at the edges of the brain, but it can be variable. So it's not very stable. Sometimes it just doesn't work at all. Yes, it is a problem, but I think as long as, I don't think that the results that I showed you in the epilepsy lesions are, caused just by differences in background field removal, because you'd expect that to affect, both hemispheres kind of equally, and also to average out across the different subjects, because not everyone's brain is exactly the same shape. So I think that is robust across, in that study. But I think yes, it's definitely a problem, and we still haven't figured out the perfect masking technique. We have some tricks that we like to use. We often try to mask based on the last echo time data set. We often try to run not only something like a brain extraction tool, but also exploit the noise map that comes from the multi-echo fitting that we do, and we try to remove the noisy areas. So we really try to do as good a job as we can on the masking and try to minimize the number of eroded voxels, but obviously it is a problem for cortical QSM.
Great. Thank you again for your talk.
It's a pleasure. Thank you for inviting me.
Let's keep in touch and perhaps run something together. Let's
That would be great.
discuss